Good afternoon, and for some of you, good evening. I'm Kathleen Stephenson, the GIC Board Chair, and I would like to welcome you to today's executive briefing. So today's topic is on the human immune response to SARS-CoV-2 and the emergence of variants. To help us understand this rather complex subject, we are honored to be joined by Dr. Michel Nussenzweig. He's the Zanvil, Cohn, and Ralph Steinman Professor and Chair of Immunology at the Rockefeller University and a Howard Hughes Medical Institute Senior Physician. He holds an MD degree from the New York University Medical School and a Ph.D. in cellular immunology from the Rockefeller University. His research aims to understand the rules that govern hypermutation and antibody selection with the goal of creating vaccines for pathogens such as the HIV-1. His molecular biology laboratory studies the molecular aspects of the immune system's responses using a combination of biochemistry, molecular biology, and genetics. He has published extensively and has received numerous awards and research achievement recognition around the globe. He's a member of the American Academy of Arts and Sciences, the U.S. National Academy of Medicine, and the U.S. National Academy of Sciences. Now, in interest of time, I will refer you to the website of both the Rockefeller University as well as the GIC for a complete description of Professor Nussenzweig's research accomplishment and distinguished career. As a reminder, this event today is being recorded and the replay will be available on the GIC website, which is www.interdependence.org. So, with no further delays, Professor Nussenzweig, the virtuous floor is yours.
Thank you, Kathleen. All right, so what I'm going to do today is I'm going to speak a little bit about the immune system, about how my laboratory got involved in this coronavirus problem, and then something about what we know about the response to coronavirus, to vaccines, and the variants. All right, so just to start, I want to do a brief introduction to the immune system because it's not something that we all think about all the time. So what the immune system has to do, its job, is to distinguish between our own bodies and everything else. Everything else means microbes. It means things that are foreign to our bodies, like cancers, and all categories of microbes and chemicals, for that matter. And to do this, the way the immune system, the properties that are essential to the immune system are that it has the specificity to see all of these different things and not mix them up. Diversity, meaning that, again, all of these different things can be seen. And memory is the property that allows vaccines to work. That is, that you're exposed to something once, that you remember it and do better the next time. So the way one can think about this, a simple way to think about it, is a construct, a theoretical construct that was outlined by Burnett and Lederberg and Talmage about 60 or 70 years ago. And it's rather simplistic, but it's the way, in fact, that I like to think about immunity. So what they postulated then was that the immune system would be composed of cells, and that each cell would have a unique receptor. So that would account for diversity, because then you'd have all of these different cells, each one with its own receptor. and the receptors would have the specificity required to recognize the foreign substance. And then memory in this construct is rather simple. It's that when a virus, for example, enters the system, there's a cell that recognizes it because it has the right receptor for that virus. And when that happens, that cell undergoes clonal expansion. It grows so that memory can be accounted for, because when you see the thing the second time, well, you've got, instead of having a single cell that you can have to find in order to do this, well, you've got lots of cells. So you have a bigger, faster response. All right, so that's a very simple model for the immune system. And how do you make all of that diversity? Well, you can imagine that if every single compound that exists in the natural world is something that you have to recognize, there are millions, in fact, of different things that you have to be able to see. And our genomes are too small for that. So the way that the immune system generates diversity is that these receptors, the antibodies, which are also the receptors on the cells that I talked about in the earlier slide, the antibodies are made up of gene segments that are brought together randomly as a lymphocyte, which is the cell in the immune system, develops. So every cell has a unique receptor from a limited number of genes that's put together randomly by gene splicing as the cell develops. Okay, so that's how you get more receptors than you have genes for. All right. And then there's another second diversification reaction that happens in the immune system. So when we get a vaccine, for example, and this is obvious from the corona experience, you get vaccinated, you get an initial response which is not that great, but then within a short time, you get a better response. That's something that was also discovered about 60 years ago. The reason that this happens is something that's really unusual that the lymphocytes do. So when they get this first exposure to the virus, let's say, and they start growing, they also start diversifying further. So they begin to express an enzyme that makes small mutations in the antibody genes. And instead of a monomorphic collection of cells that all have the same receptor, what you get is a pleomorphic group of cells that differ from each other by just small mutations in the antibody. So a further diversification, but aimed at the cell that's recognizing the antigen. And during the immune response, cells that have the best antibodies are selected. Okay, so that's how you get a second diversification and selection during the immune response. And that happens in specialized structures called germinal centers. These are something that histologists have looked at for well over 100 years. And this germinal center has two products. It produces memory cells. So that output that we talked about earlier, the expanded clone, that's the memory cell, is put out from this germinal center, as are cells called plasma cells, which are antibody factories. So two different compartments, a memory compartment, which sticks around, and a plasma cell compartment, which produces the antibodies which are in circulation, which are going to be protective. All right, so plasma cells are very highly selected in the way I diagrammed earlier. Memory cells, on the other hand, are different, and that's going to be important for talking about coronavirus. Memory cells are, in fact, a diverse collection of cells that carry some of these mutations that change the antibody just slightly. All right, so My lab became interested in human immunity about a dozen years ago when we tried to understand how some people that are infected with HIV actually make good antibody responses that could be protective. But these are very rare individuals. And so we developed methods to try to understand what was happening in those people. And the method that is relevant to coronavirus is to find the cells in the blood that make antibodies. So here are the cells, the B lymphocytes that have the antibody on the surface. Find these cells and see what they're making. And the way we do that is by taking the protein that is of interest. In the case of SARS, it's the SARS spike protein, and label these lymphocytes from blood with the protein. They can capture that protein specifically because they're making the antibody that sees that protein. And then we sort single cells and do molecular biology to clone out their antibody genes. It sounds complicated, but it's something that we can teach a high school student in a couple of months, really. The other thing that's important for coronavirus is to have an assay that will allow us to measure neutralizing activity. And the way we do that is we create a pseudovirus, a virus that has in it a luciferase gene, a gene that emits light that we can measure, and also carries the SARS protein on its surface, the spike protein. And that kind of assay has a huge dynamic range, several logs, and it is really very sensitive. So when the coronavirus epidemic hit New York City, and in fact, even before that, in January, we began recruiting people that were infected to try to understand what their antibodies were and how people respond to this infection. And I'm just going to show you something about what the neutralizing activity is in a group of 150 people. So these are, each dot represents a person, and this is their neutralizing titer here. And you can see it's a log plot, so there are big differences here. And these are the top 60 or so individuals that are depicted here. So, these people down here, which are 50% of the cohort, are already having pretty low antibody titers. And fully 30% of the individuals that we assayed, and this is not just our own findings, but everybody pretty much that's done this, about 20 to 30% of people don't really have a measurable antibody titer after they're infected with the virus. That's important because it's the antibody that's going to be protective. All right, so 6 months later, what we find in these same people is that they've all dropped their titers. They've dropped their titers by about 5-fold on average. So even here, you can start seeing people where we cannot measure neutralizing activity. And of course, the ones that are lower are even worse. Good news is that after a year, it's pretty much the same as it was after 6 months. So what's circulating in plasma in people is retained really for quite a long time after the infection. And people who are vaccinated and had been infected, of course, are doing exceptionally well. And those are the red dots. Okay, so what about vaccines? So what I just told you was about natural infection. And here are some vaccine data. So again, this is the neutralizing titers here. This is the group at the first time point, which is a little bit over a month after they were infected. This is after 6 months, so a significant drop in titer. And here are people that were vaccinated with either Moderna or Pfizer, and they look pretty good. So they have pretty good titers against the virus. It's the same for Moderna and Pfizer. But unlike the naturally infected people, these people are losing their titers at a more rapid rate, so that 2 months after the second injection, they're already down by a factor of 10. And we're going to see what they are at 6 months, but we really don't know yet. Now, that's one compartment. That's the plasma cell compartment. What about the second compartment, the memory cell compartment? And that's all important because it's going to be what keeps people out of the hospital and keeps them from having a serious illness. All right, so identifying those cells, again, is a matter of using the spike protein, taking blood, using the spike protein to identify those cells, and then sort them as single cells, and then do molecular biology. And this is what it actually looks like when you do this. So here, each column is an individual. These are 6 people that we've looked at. 4 of them we've looked at 1.3, 6, and months and a year. And what's in here is the memory cells. And you can see that they're rare in terms of the percentage of total B lymphocytes, but they are steady. So you can see that they're not really changing even after a year. And that's really good news for people that have been infected with the virus. And here we're looking at the vaccinees in this column compared to the naturally infected. And you can see that the vaccinees too have a substantial memory compartment. So that's all very good. Now, when we look at the antibodies in these people, one of the striking findings is that every person that we've looked at, and these are Moderna Pfizer vaccinees, each one of these bars is a person, let's say. And here are the naturally infected in green. And the lines in this so-called circus plot are identities, antibody identities. And so what you can see here is that there are very, very common antibodies present in every single person that was either infected or vaccinated. That's a rather surprising finding given the diversity that I talked about earlier. So you can create a lot of diversity in the antibody system, but despite all of that diversity, people are making very, very similar responses to this virus. All right, so I'm just going to summarize this. This is really a lot of information here. So infected individuals retain serum neutralizing activity. Actually, it's after a year, but it's decreased about 5-fold from the peak. There's a lot of diversity there. There are people who have very low levels of neutralizing activity. The memory compartment, memory B cells persist even after a year. The mRNA vaccination and natural infection elicit similar levels of neutralizing activity. Older individuals, I didn't show you this, produced far more modest responses. So there's a linear correlation between age and vaccine response. Vaccination prevents serious disease, but it does not prevent infection. Again, this is data that I haven't shown you, but in fact, people who are vaccinated and might have those low levels of antibodies in plasma or serum can be reinfected, but they don't seem to be getting serious infections because of their memory responses. The half-life of the mRNA-elicited antibodies appears to be shorter than actual infection. This is something that we're going to have to keep an eye on, and people will likely have to be boosted with these vaccines. Good news is that the vaccination does elicit antibodies that are very similar to natural infection and that are quite effective at neutralizing the virus. All right, so how can we explain some of these biological phenomena? Well, we learned a lot about antibodies by essentially taking their pictures by crystallographic means, nowadays electron microscopy. This is a representation of the protein that's on the surface of the virus that gets it into cells. And the bit that gets it into cells is called the receptor binding domain, in red here. This is a trimeric protein. It means it has 3 identical parts. And the cellular receptor that it binds to, called ACE2, is depicted in green. The way we did our sorting, we used this little bit, the red bit, as a bait. And the reason we used the red bit as a bait is because we felt that if we could find antibodies that bound to this receptor binding domain, it would interfere with its binding to the cellular receptor and be neutralizing. And that's exactly what we got. And we also got an understanding of why it is that everybody is making the same antibodies and how they block the virus. So here is an actual structure of the receptor binding domain and the actual structure of an antibody binding to it. And the way that this antibody binds to it is exactly the same way that the ACE receptor on the cell binds to it. So it blocks that interaction. And it blocks that interaction using bits and pieces that are encoded in the genome, as opposed to VDJ recombination. And because everybody has very similar bits and pieces in the genome, in their genome, then it's easy to understand how everybody makes the same antibody. All right, so our antibody cloning efforts led to the finding that there are 4 dominant classes of antibodies that are neutralizing, that bind to this receptor binding domain, and they're shown here. Some of these are part of, for example, the Regeneron combination that's in clinical use. Others are also the Lilly antibodies, and the recently acquired Bristol-Myers Squibb antibodies are all depicted here. The importance of finding these different types of antibodies is that you can see that some of them bind to different areas. So class 1 and class 2 are overlapping classes, and they're distinct from these other 2. So if you are worried about, for example, the emergence of one particular variant, well, then you might use combinations of antibodies that target different areas in order to avoid loss of activity. And that's exactly what's in the Regeneron combination. The Regeneron combination uses class 1 and class 3 antibodies. All right, so what about the variants? So what you can see here is, again, the depiction of the receptor binding domain with the amino acids that are the emerging variant amino acids. And the key emerging variant amino acids are these 3, 417, 484, and 501. That's what's in the South African variant. That's what's in the Brazilian variant. 484 is in the New York City variant, and 501 is in the British variant. So you can understand from looking at this picture and where these things are that they might interfere with the antibodies. And in fact, that's exactly true. So here is an example of a class 1 antibody binding to the receptor binding domain, and you can see these residues, the residues that are implicated as important in emerging variants, just underneath the antibody. And in this class of antibodies, in fact, it's 417. If you mutate 417, they lose activity. Or if you mutate 501, many of them lose activity as well. A second class, class 2, shown here, is very dependent on the 484. And you can see that several different antibodies in that class are all lost activity against the 484 mutation. So it's not surprising then, and these two classes, class 1 and class 2, are the dominant classes in serum, and they represent many of the antibodies that are shared by all individuals. So then it's not surprising that if you look at convalescent people at 1 or 6 months, and their ability to neutralize the wild type versus the mutants, that they lose activity against the mutants in both cases. And vaccinees also lose activity against the mutants because they're all making the same antibodies, and these antibodies are susceptible to these mutations. Now, just saying they're susceptible doesn't mean that the mutations were essentially a response to the antibodies or selected by the antibodies. But in fact, we believe that that's exactly what happened. So, and the reason we believe that is because we essentially predicted exactly the mutants that are coming up using an accelerated evolution experiment. And I'll just tell you exactly how that works. We have a virus, which is not SARS, but in which we express the SARS spike protein, that trimer that helps it get into cells. We take that virus, which has a higher level of mutation than the SARS virus, and culture it in the presence of one or another of the antibodies, and ask, does the antibody select for the mutants? And that's exactly what we see. So this is a collection of about 20 different antibodies in the different classes. And here you see exactly what they select for. So they select for 417 mutations. They select for 484 mutations. That's the class 2 that's selecting for that. And they also select for 501. So they select for exactly the mutations that are coming up in the variants. All right, so vaccinee and convalescent plasma have reduced activity against the variants. That's not at all surprising given what kinds of antibodies people are making. The monoclonal antibodies that you derive from these people are sensitive to these mutations, and antibodies are in fact selecting for the mutations. That's what's happening in vitro, and that's what we believe is happening in vivo as well. All right, so just a word about antibodies and therapies or prevention. So antibodies can be engineered to have very long half-lives, such that they could be administered once or twice a year. They're protective in animal models, and they are protective in the clinic as well. So individuals that cannot respond to vaccines, so all kinds of immunodeficiencies, could be administered antibodies for protection, but that's a small segment of the population. Additionally, antibodies are also effective as therapeutics if administered early in the infection when people are viremic. That's also clear from several clinical studies now. All right, so I just want to summarize, and then I'm happy to take questions. What we've been doing is studying people that are either naturally infected or vaccinated. We do it at Rockefeller University, where we have a very small hospital which is dedicated entirely to clinical studies, such as the ones that I've been talking about. We do a lot of this by analyzing the kinds of antibodies that people are making, and we do this by techniques that we developed and that are now broadly available. And all of the antibodies essentially that are in the clinic come from this kind of technology. We've discovered that everybody's making very similar antibodies, and the fact that people make very similar antibodies partially explains why and how we see the emergence of variants, the same variants in many different parts of the world. And it's because of the parts of the virus that are being targeted by the antibodies. It's not just that they target these areas, they also select for the variants. Okay, and I just want to say that what I've summarized is the work of many, many people, and actually very collaborative work, scientists that are not just at Rockefeller University, but all over the country, basically, including labs at Caltech and UNC and Mount Sinai and all kinds of places, really. So I'm happy to take questions if there are questions.
Can you hear me?
Sure.
Yeah. So thank you so very much. I mean, this was quite an amazing presentation. You know, when you use the word easy to understand, I kind of like, maybe not. But anyways, while we're waiting for questions coming in, I just want to ask you one, which is, you know, at the end of the day, there appears to be a cyclicality here in the pandemic. You've got, you know, the surge, the plateauing, the emergence of new variants, and then the responses, the tweaks in the vaccine, the therapeutic research, and mask wearing. And that's in response to, obviously, these changes within the cycle. Do you think that this dynamic is the new normal for years to come? Because that would, if so, that would have tremendous implication not only on the day-to-day life of people, but you know, the economics. I mean, industries, the economy at large. So I think that's one thing that you know, I would like.
What's the future hold for us? Question. Yeah, I think the vaccines are really quite effective in keeping people out of the hospital, because they produce this group of memory cells that can respond to subsequent challenge. What that means is that we are going to dumb this thing down so that it becomes more like a cold or a flu infection, so that we are going to be able to get back to our normal lives. And that's going to depend on vaccination and boosting. So I'm very optimistic that we will be able to get back to something more like normal. Does that mean that the infection is going to go away? Absolutely not. Okay, we're going to have this thing for the rest of our lives. It's going to be part and parcel of being a human being. But you know, having the flu around every year didn't stop us from doing things. And that's what this is going to become. It's going to become a lot more like the flu, but that's dependent on vaccination. So for people that refuse to take the vaccine, or for which the vaccine is unavailable, we're going to continue as we are until they all basically get infected. And that's going to take a long time. Yeah,
yeah. I guess, and therefore the mask wearing is pretty crucial in that respect.
Yes, people should continue to wear a mask even if they're vaccinated. So I think that the CDC recommendations are correct. If you're having, you know, a few people over for dinner and everybody's vaccinated, that's fine. But in going out into go do your shopping at the supermarket or whatever, you wear a mask.
Yeah.
Because even if you're vaccinated, you are going to get infected, assuming that your plasma levels are low, especially people that are my age and above. I'm 66.
So we are inundated with questions. So I hope that you have nothing to do in the next 4 hours. One is with regard to two questions about, did the virus become a pandemic, and how did the virus become a pandemic? And what is the most important thing that we can, do to reduce this impact?
Vaccines and masks, that's it. And how does it become a pandemic? Well, it's people. That's the reservoir. We're the reservoir, and probably some, small animals are also reservoir, but we're the important reservoir. So when you go to the opera, you're sitting there in a sea of reservoir. Better wear your mask.
Yeah, yeah. That's for sure. And we can assume that we will need these booster shots all the time, right?
The current vaccines, I believe, will require us to be boosted. And the good news is that, these vaccines, the messenger RNA vaccines, are easily modified so they can take care of these variants. And the other sort of, I think, really good thing is that although we see variants emerging, we're seeing some very, very similar kinds of variants coming up everywhere. So let's say if the virus was more like HIV, where there's a great deal more plasticity and you can have a tremendous amount of variation and still be a viable virus, we would be in deep, deep trouble. But what we're seeing instead is that the virus is finding the same solution, same exact solution. So it's not just the same amino acid that's being modified, but it's being modified in the same way in many of the variants. So the virus doesn't seem to have a lot of space, which is really good because we can sort of catch up to it.
Right, so that means that it reduces, in fact, the time for a vaccine to adapt to any mutation.
Yeah, it's right. I mean, I think we're lucky about the diversity problem in the virus.
So other questions is that the warp speed, if you could tell us where the warp speed program enabled the fast introduction of effective vaccines.
Yeah, I mean, I don't know all that much about that program other than, it really put a lot of money into the vaccine developers, allowing them to do this without taking a huge economic risk, right? So normally companies don't do this at risk, right? Because what are they going to get out of it? But what Warp Speed allowed them to do was to take that capital and use it for this purpose.
Right, right. I mean, this is one of the perfect examples, I guess, where the public sector is helping out the private sector, which it would have taken a much longer time had it been just for the private sector to do this.
Yeah, I think it's actually an amazing example of how that worked well.
Yeah, yeah. There is a question about the vaccine strategy, which, if there is such thing, that could produce a variety of different antibodies, let's say, in anticipation of these mutation, given that the mutation, as you mentioned, are essentially, relatively small in numbers.
Yeah, the fact that that these variants have emerged everywhere and they're very similar will allow the vaccine developers to produce vaccines that that carry these mutations. And it's very likely that so if you are primed, let's say, with the wild type virus, and you produce memory cells that have all those little mutations that I talked about, some of them will actually see the mutant. So you're going to then have a collection of cells that are both to the wild type and to the mutant if you're boosted with the mutant, and be in really quite good shape. So I think that will work quite well.
And another question is, if you could comment on the potential antiviral therapeutics. Do you see any role for that in the approach to the pandemic?
The antiviral therapeutics right now, the best thing is antibody. It really is effective, especially if given early, and then steroids if given later. But that is for people that are really very ill. So what the companies that are developing these things are aiming for is something that you can take as soon as you're diagnosed, relatively early, that will prevent you from going down, a poor outcome path, or just something that will get you back to work sooner and make the disease less onerous. But antibodies right now are the closest thing in the horizon. There are small molecule drugs that are being developed, but they may take quite a bit longer to get there.
So there are a couple of questions with regard to the the vaccines themselves. One is it too soon to tell anything about the J&J vaccine? And another is, there's a lot of controversy now surrounding the AstraZeneca. I mean, can you comment on those?
Well, J&J is approved, right? And I think it's probably quite a good vaccine, actually. I don't know, and nobody knows really, how well that will compare to the messenger RNA vaccines in terms of how long lasting it will be, or what effects it has on memory and persistence of memory and so on. Well, that's something that we're going to have to take some time to learn about. The AstraZeneca, that's the cheapest of all of them. It's the one that's being produced all over the world. The one that's most available. I don't know that there really is a problem with it. The number of people that have had this, clotting issue is small, and it's not clear to me that that's any different from what would happen with that number of people anyway. So it's possible that there's a problem, but if it is, it's a rare event.
So it's been blown up a little bit in the press, or the the reaction of the countries of stopping administering that particular vaccine is.
It's the least immunogenic of the vaccines. Okay, so it is not the best of the vaccines, certainly in terms of the immune response that it produces, and I think you just have to weigh that off with the fact that it is going to have an effect in keeping people out of hospitals. It will have an effect on the pandemic, but in the best of all possible worlds, you'd use maybe the Russian vaccine instead. It's probably better, or J&J, or one of these other ones. Yeah. It's not the best vaccine.
And there is a couple of questions with regard to the transmission. A vaccinated person can be transmitting the virus. I mean, can you update us a little bit on that?
That's definitely the case. So, what I mentioned was that there are these two compartments. One is the plasma cell compartment, and one is the memory compartment. So if you are vaccinated, you have a memory compartment, which is sitting there waiting, and you have a plasma compartment, which is circulating. If your plasma compartment is low, which it is in a lot of people, you will get infected. You won't fight off the virus, you will get infected. But when you get infected, your memory cells will get in there, do their job, and save you from having a horrible outcome. Now, that means you're going to get infected. That means you're going to be able to transmit.
So it's through the infection and then the transmission. So at the end of the day, I mean, can you be asymptomatic and transmit anyways?
Absolutely. Absolutely. And about 40% of individuals that are infected are asymptomatic.
Interesting. Yeah.
They don't even know they're infected, but they're spreading it around.
Right. Yeah.
Yes, lovely.
So, and how big is the risk that one of the variants could be really nasty, lethal?
It doesn't seem to be the case. What seems to be the case, and this is really kind of not, to my mind, not very well established, is that some of the variants give you a slightly higher viral load, right? That's, if they give a slightly higher viral load, you may have a higher probability of having a poor outcome. But the numbers are just, you know, not very believable on that.
So there is a question about the fact that, I mean, I'm not sure whether the person who's asking the question is skeptical about the vaccine, but, you know, to what extent, when do you expect there will be some reliable information about the duration of protection afforded by the current vaccines?
You know, we don't know. I think if we have a 1 year time between vaccinations where it remains effective in the sense that it keeps people out of hospital, then we're doing really, really good. You know, people get vaccinated for influenza once a year. What's wrong with getting a second shot for coronavirus?
Yeah, yeah. So there is I think a public policy that needs to be much more effective in terms of explaining that this is a you know, a new normal situation in a way, and I think that I think there is a disconnect, right? Because you've had such high mortality. And, you know, now you've got a vaccine and people are, you know, still thinking, oh, they're going to inject this particular virus in me. I mean, there's a lot of that kind of belief still, just like
Yeah, everybody has to be educated. I think it's really good what the current administration is doing, which is they're going to spend a lot of money on a public campaign to try to explain why people should be vaccinated, why the vaccine is safe, and so on. It's clearly as important as having one as getting people to take one.
Right. And at this point in time, is there any benefit of having a cocktail of vaccines, or it doesn't matter at the end of the day?
No, I don't think we know enough about that. No. I know, for example, is it better to get vaccinated with Moderna and then come back with Johnson, or vice versa, or just continue with what you started with? And all those things will be worked out in the future.
Yeah, yeah. So, chance of broad spectrum. So there's one question with regard to the COVID, stressing the importance of moving the mRNA vaccine, both for the speed, because they have better chance of being a more a broader spectrum, if you will. Can you tell us how that works?
Yeah, I mean, the mRNA vaccines are faster to modify because the process for, you know, chemically the process is reproducible and, you know, highly predictable. When you make a new protein, on the other hand, it's far less predictable whether or not you're going to be able to make that protein or that virus. So the mRNA vaccines have that as a major advantage. However, they may not be as long-lasting as some of the other vaccines. And so they are probably best thought of, and they're very expensive, and they're a pain because they have to be kept at certain temperatures and all of that kind of stuff. So ultimately, they may fall away in the long term. And something like the Johnson vaccine or a Novavax or something else like that, that takes a little bit longer to develop, may be the better option. Simply because who's got a -80 freezer in the pharmacy? And why do you need 2 shots? And will the levels fall away faster with the messenger RNAs, which I believe they probably will. So it's going to be the stopgap, the thing that you can do quickly and modify quickly, but it may not be the one that you want in the long run.
Well, that's interesting. So in other words, this new technology is, you know, very seductive in a way, and probably adaptable to other things, but you think that, you know, these challenges are such that it limits it in the long run?
I think it may be more limited than the other options in the long run. But you know, we'll see.
And how optimistic are you to develop, you know, therapeutic drugs that, you know, will be equivalent to the Tamiflu, let's say?
Yeah, I think that will happen. But developing a small molecule drug is a lot more difficult than the antibodies. The reason it's more difficult is because the antibodies are coming out of people, right? So they're coming out of infected people. They've been filtered through us. So we know they are safe for us. They are a natural product, right? It's from one human to another, basically, with a cloning step in between. The small molecule drugs are things that you create in a laboratory, and they have all kinds of issues related to safety because they haven't been filtered through a person. Pharmacokinetic properties, manufacturability, et cetera, et cetera, et cetera. So there's a whole long list of something that you're creating that you don't know whether or not it's going to work in a human. And so going from something that works in a test tube to an effective drug, it takes quite a lot of doing.
So it's more the, I'm not sure I entirely follow. So it's more the production of, you know, and the safety of the production than the constraints of production of taking antibodies from. Yeah,
let's say, look, I mean, one way to explain it is like this. If I make an antibody and it's the same molecules that you would make, I can give my antibody to you and I know it's going to be safe. It's not going to be an auto antibody. It's not going to create a problem in your body because I know that because it was already in a person, right? Now, if I create a molecule in a test tube that I know that molecule will inhibit the SARS virus, I don't know that that molecule will also, you know, create a problem in your brain. It could, okay? So just going through all of the steps that are required in order to make sure that something that you create in a test tube as a chemical is safe and effective for human use is a whole to do. You know, that's really takes quite a lot of doing. As opposed to an antibody, I basically, as soon as I get it, I know it's going to be good.
But the harvesting of antibodies then is a constraining element, isn't it?
The hardest thing about antibodies is that they're more expensive and they have to be injected. Right. That's the hardest thing. You know, at the current rate of production, a dose of the antibodies, for example, that Bristol Myers are going to produce probably costs $40 to make and stick in a vial, right? So the $40 to make, of course, they're going to charge $2,000 or whatever they're going to charge, or $1,000 or $500, but they're going to charge something more than that. A small molecule may cost a penny to make. They're still going to charge $500 or $1,000, but you know, it costs a penny to make.
Yeah. So we're back to to that equation, maximizing profits. So one last question here, and that is, do you believe that there is a possibility for us developing a way of fully fighting and eradicating the virus? And I think you did answer that earlier, that it's...
I don't think so.
Yeah.
I don't think so. If we can't eradicate flu, I don't think we're going to eradicate this. We're not going to eradicate the common cold. You know, we can make it into something that's tolerated by our society, the way we live, but getting rid of it, it seems unlikely.
Yeah. And do you think that, you know, by and large, we have, you know, the society, and I think the policy responses have learned a lesson with this pandemic for a potential new pandemic in the future?
I hope so. You know, I hope that the way that what we've learned here will help for the future, and that there will be additional investment in trying to have an even more rapid sort of response for the future, because we will have other pandemics. This is the third emergence of something like this within, you know, a couple of decades, 2 or 3 decades. Does that mean that we're never going to get another one? No, it means we're going to get another one.
Yeah. 2 observation makes a trend already. So it's, well, Michel, thank you so much. I think that we are coming close to the end of your presentation and the discussion. I really want to thank you again. And, you know, in a way, I hope that maybe you could come back and update us on the recent developments and so on. But again, it's we've learned, I think I certainly have, and I'm sure the audience also have learned quite a bit on all this debunking of the variants and the vaccine. So thank you again for your time.
My pleasure, Kathleen.
Take care.
Bye for now. Bye-bye.
Goodbye. And as a reminder for the audience, before you leave us, I'd like to just mention that the briefing will be available on the GIC website as soon as tomorrow. And then turning to our next executive briefing, happens on next Tuesday, March 23rd, and the subject there will be very, very different. It's a special College of Central Bankers presentation that will explore monetary policy and its impacts with Anna Paulson, who is the Executive Vice President and Director of Research at the Federal Reserve Bank of Chicago. And she will be talking with former President and CEO of the Federal Reserve Bank of Atlanta, Dennis Lockhart, who is also a GIC College of Central Bankers Fellow. So today's seminar is part of the David R. Kotok Global Healthcare Series, which David had established with a very general matching donation campaign, which goes on through this month, the month of March. All donations go directly towards bringing high quality content as a public service for the remainder of this year. So to find the video replays, to explore the GIC membership, and perhaps more importantly, to donate to the GIC, you can visit our website at, again, www.interdependence.org. As we conclude, I might ask you for another 60 minutes of your precious time, and that is to respond to our survey that you will get when you exit this webinar. And with that, I want to thank you for your attention and wish you a very good evening. Bye-bye for now.